Hi everyone, and welcome to today's lesson on sex-linked disorders. In this video, we're going to break this down step by step so that by the end you actually understand it and don't just memorize it. Sex-linked inheritance refers to traits that are controlled by genes found on the sex chromosomes. This sex chromosomes are usually found on the 23rd pair. Sex linked disorders are especially found on the X chromosome. In humans, we have two sex chromosomes. Females are XX and males are XY. Now, here's an easy way to think about it. The X chromosome is like a big textbook full of information. The Y chromosome is more like a small booklet with much less information. Sex linked inheritance focuses on traits linked to genes on the X chromosome. Now, here's where it gets interesting. Males only have one X chromosome. So, whatever is written in that textbook, they have no backup copy. One example of a sex linked disorder is hemophilia. This is a condition where the blood does not clot properly. So, even a small cut can lead to a serious bleeding. To represent hemophilia, we use symbols. X capital H is normal, X small h is hemophilia. Now, remember, the Y chromosome does not carry this gene, only the X chromosome does. This means males are more likely to show these disorders. Remember, females have two X chromosomes, so it's like having a spare copy. If one has a mistake, the other one can often cover for it. But this is not true for males. Males only have one copy of the X chromosome. A carrier female has one normal allele and one hemophilia allele. She does not show the condition, but she can pass it on. It's a bit like carrying a hidden message that can be passed to the next generation. An affected male has one X with the hemophilia allele and one Y. There's no second X to fix the problem, so the condition is expressed. When solving genetic crosses, always follow these steps. First, write the genotypes of the parents, then determine the gametes. Next, draw your Punnett square and finally work out the offspring. The Punnett square shows all possible combinations. Think of it like a prediction grid, showing all the possible genetic outcomes. From this, we can see who will be affected, who will be carriers, and the chances of each outcome. This is where exam marks are often earned or lost. So take your time here. Remember, sons get their X chromosome from their mother and their Y chromosome from their father. So fathers cannot pass X linked traits to their sons. That's a very important pattern to remember. Let's quickly summarize. Sex linked traits are carried on the X chromosome. Males are more likely to be affected. Females can be carriers. And genetic crosses help us predict inheritance. Now, before you go, make sure you watch the Notebook LM generated video at the end. This will help you reinforce what you've learned in a slightly different way. Sometimes hearing the same concept explained differently is exactly what makes it click.
You read the genetics problem, you draw the boxes, you plug in the X's and Y's, and you still get the wrong percentage. We are going to build a step-by-step framework to read the prompt, set up the cross, and pull out the exact percentages every single time. Grab a pen and some paper. You will need to write this down to build the muscle memory. By the end of this, you will have a perfectly structured, flawlessly labeled Punnett square that calculates exact inheritance percentages. Success in genetics depends on identifying the correct rule set before you draw a single box. The absolute first thing you must do when you look at a genetics problem is scan the text for where the gene is located. In this word problem about cystic fibrosis, the first sentence specifies that the recessive allele is carried on chromosome number 7. This location determines your entire approach. Any trait carried on chromosomes 1 through 22 is an autosomal disorder. It impacts males and females equally, so we use standard letters to solve it. No X's and no Y's. Compare that to the problem about Jensen syndrome. The text identifies it as a sex-linked disorder and states the allele is carried on the X chromosome. This flowchart maps out your exact decision process. If the trait is on chromosomes 1 to 22, it's autosomal, and you use basic letters. If it's on the 23rd pair, it's sex-linked, and you must use X and Y chromosomes in your cross. Applying X and Y to every single problem you see is a common cause of failure. You have to verify the location first. Let's apply those sex-linked rules to the Jensen syndrome problem and write out the starting genetics for our parents. This table breaks down the possibilities. For males, there are only 2 options. Because they carry only one X chromosome, they are either normal or they are affected by the disorder. Females have 2 X chromosomes, which creates a third option. They can be normal, affected, or a carrier. A carrier hides the recessive allele on one X chromosome, but remains physically unaffected because of the dominant normal allele. Our problem tells us we are crossing a normal male with a carrier female. To keep the information organized, we use a specific layout called the recipe. We list the P1 phenotype, which is their physical description, followed by the P1 genotype, their exact alleles, and then a step for meiosis. During meiosis, we split those parental alleles apart. Each sex cell only gets one chromosome, so we physically separate the letters on the page to reflect that biology. Next comes fertilization. We draw a 2x2 grid. The mother's separated alleles go along the top columns, and the father's alleles go down the side rows. Now we combine them. In the top left box, the 2 normal alleles meet, giving us XB, XB. In the bottom left, the mother's normal allele meets the father's Y chromosome. In the top right, the mother's recessive allele pairs with the father's normal allele. Finally, in the bottom right, the mother's recessive allele pairs with the father's Y chromosome. Following this checklist turns a complex biological concept into a predictable data sorting task. These 4 boxes represent the 4 possible outcomes for this couple's offspring. We have our raw data, but we aren't done until we translate these combinations back into physical descriptions. Genetics prompts often use specific phrasing that can lead to incorrect calculations if misread. Looking at the results, we have 1 normal female, 1 normal male, 1 carrier female, and 1 affected male. If the question asks for the chance of an affected son, look closely at your grid. Out of the 4 total possibilities, only 1 is an affected male. That makes the answer 25%. However, if the question asks for the chance of a normal child, regardless of biological sex, we count both the normal female and the normal male. That is 2 out of 4, making the answer 50%. Precision in reading the final sentence of the prompt is just as critical to your grade as the math inside the Punnett square. And there it is. You just completed a perfectly labeled, correctly calculated sex-linked genetic cross. Look at this entire structure. You avoided the autosomal trap by checking the chromosome location, you laid out your starting data using the recipe checklist, you executed the grid, and you pulled the exact targeted percentage requested. To make this permanent, take a blank piece of paper and practice this exact sequence 3 more times with new prompts. Lock the workflow in, and you will never miss a genetics question again.